Welcome back to the channel where medical topics are made easy. Today we're discussing gram-negative bacterial infections. Make sure to watch until the end because at the end I have a question for you. So put your thinking cap on. We're going to start with Pseudomonas. Pseudomonas aeruginosa is an opportunistic aerobic gram-negative bacillus that is a frequent deadly pathogen of people with cystic fibrosis, severe burns, or neutropenia. It is an aerobic bacterium found in soil and water, and it grows easily on non-nutrient media. You can think of this microorganism as a bit of a bully.
Ho, ho, ho!
It does not bother healthy people unless they have some sort of predisposing condition such as cystic fibrosis, burns, neutropenia, diabetes, and so forth. For example, many people with cystic fibrosis die of pulmonary failure secondary to chronic infection with P. aeruginosa. They also commonly infect debilitated patients in hospitals, hence its reputation for being a hospital-acquired pathogen. P. aeruginosa can be very resistant to antibiotics, making these infections difficult to treat. It often infects extensive skin burns, which can lead to sepsis.
Ho, ho, ho!
And it can be transmitted in healthcare settings via contaminated hands, equipment, etc. It has been cultured from wash basins, respirator tubing, nursery cribs, and even antiseptic-containing bottles. It also causes corneal keratitis in wearers of contact lenses. If you have ever worn contact lenses, then the risk of developing microbial keratitis is 450 times higher if you're not careful about hygiene. But hey, when you're looking to make a big impression on someone, all you need to do is get them a pair of contacts and boom. Now they're 450 times more likely to develop a corneal ulcer.
Ho, ho, ho!
And no, that joke was not lost on me. I'll stop now. It causes endocarditis and osteomyelitis in intravenous drug abusers, external otitis or swimmers, or in healthy individuals. But it can cause severe external otitis in people with diabetes. And it can grow in the urinary tract, causing pyelonephritis. It can also grow in wounds, causing eczema gangrenosum. It loves growing on burn wounds. In fact, 70% of burn wound infections are caused by this microorganism.
Ho, ho, ho!
This organism is a major cause of morbidity and mortality in people who sustain thermal injuries, and it is the leading cause of death in patients with severe burns. As far as labs go, Pseudomonas is a lactose non-fermenter. On McConkey agar it will grow as a non-lactose fermenter, appearing as pink colonies. Other important characteristics include, it is oxidase positive, catalase positive, and it produces a characteristic grape-like odor when grown on cetramide agar. Pathogenesis. This microorganism produces several toxins that contribute to local tissue damage.
Ho, ho, ho!
The organism secretes an exotoxin called exotoxin that, like diphtheria toxin, inhibits protein synthesis by ADP ribosylation of the ribosomal protein EF2, leading to the death of host cells. Exotoxin is particularly lethal to ciliated epithelial cells lining the respiratory tract, and it contributes to the necrotizing pneumonia seen in susceptible hosts.
Ho, ho, ho!
Interestingly, Pseudomonas aeruginosa also secretes a heat-stable hemolysin called RTX toxin, which causes cellular membrane damage in neutrophils and macrophages, impairing their function and allowing P. Pseudomonas to evade the host immune system. P. aeruginosa also secretes damaging enzymes that destroy extracellular matrix elastase, kill leukocytes, leukocidin, and destroy cell membranes hemolysins. It also secretes other virulence factors such as superoxide dismutase and catalase, which allow it to resist the oxidative burst of phagocytes.
Ho, ho, ho!
an alginate capsule which helps it evade phagocytosis, flagella which allow it to both adhere to host cells and resist being destroyed by shear forces in biofilms, and it produces a slime layer and biofilm which help it adhere to surfaces such as indwelling catheters and medical devices. The organism also secretes several proteases that allow it to survive in the human body. Lastly, in the lungs of people with cystic fibrosis P. aeruginosa secretes a mucoid exopolysaccharide called alginate which forms a biofilm that protects bacteria from antibodies, complement, phagocytes, and antibiotics.
Ho, ho, ho!
The organism rapidly develops antibiotic resistance through other mechanisms as well, making treatment difficult. Signs and symptoms. Pseudomonas causes a necrotizing pneumonia that is distributed through the terminal airways in a fleur-de-lis pattern with striking pale necrotic centers and red hemorrhagic peripheral areas. On microscopic examination, masses of organisms are seen that tend to be most concentrated in the walls of blood vessels where host cells undergo coagulative necrosis.
Ho, ho, ho!
This picture of gram-negative bacterial vasculitis accompanied by thrombosis and hemorrhage, although not pathognomonic, is highly suggestive of P. aeruginosa infection. One of the main complications of Pseudomonas in the lungs of people with cystic fibrosis is bronchial obstruction caused by mucus plugging. This mucus provides a perfect environment for Pseudomonas to grow, leading to subsequent infection and inflammation. Chronic Pseudomonas infection despite antibiotic treatment and the host immune response may result in bronchiectasis and pulmonary fibrosis.
Ho, ho, ho!
In skin burns, Pseudomonas proliferates widely, penetrating deeply into the veins and spreading hematogenously. Well-demarcated necrotic and hemorrhagic oval skin lesions called ecthyma gangrenosum often appear. Disseminated intravascular coagulation is a frequent complication of Pseudomonas bacteremia. Now here's a quick review of the other gram-negative bacteria. Enterobacteria are a diverse group of bacteria that reside in the gastrointestinal tract of humans and animals. These bacteria are typically classified based on their ability to ferment lactose.
Ho, ho, ho!
In general, enterobacteria are categorized as either lactose fermenters or non-fermenters. Lactose-fermenting bacteria such as E. coli and K. pneumonia are able to ferment the sugar lactose-producing acid and gas as a result, whereas lactose non-fermenting bacteria such as Salmonella and Shigella cannot ferment lactose and do not produce acid or gas. Enterobacteria can cause a wide range of diseases, from mild gastroenteritis to severe systemic infections.
Ho, ho, ho!
Some examples of enterobacteria and the diseases they cause include coli, which can cause urinary tract infections, pneumonia and gastroenteritis; Klebsiella pneumonia, which can cause pneumonia, urinary tract infections, and meningitis; Proteus mirabilis, which is commonly associated with urinary tract infections; Enterobacter species, which can cause wound infections, urinary tract infections, and pneumonia; Serratia marcescens, which can cause wound infections, urinary tract infections, and meningitis;
Ho, ho, ho!
Citrobacter species, which are often associated with urinary tract infections and sepsis; and lastly, Yersinia pestis, which is the causative agent of bubonic plague. Vibrio species are a group of gram-negative bacteria that are typically found in aquatic environments. The most well-known species is Vibrio cholerae, which is the causative agent of cholera, an acute diarrheal disease that can be life-threatening if left untreated. Other Vibrio species, such as Vibrio parahemolyticus and Vibrio vulnificus, can cause foodborne illness in humans.
Ho, ho, ho!
Helicobacter pylori is a spiral-shaped gram-negative bacterium that is a common human pathogen. It is the causative agent of gastritis an inflammation of the stomach lining that can lead to peptic ulcers and stomach cancer if left untreated. H. pylori has a unique ability to survive in the highly acidic environment of the stomach, allowing it to colonize the stomach lining and cause disease. Bartonella henselae is a gram-negative bacterium that is commonly found in cats and is the causative agent of cat scratch disease.
Ho, ho, ho!
This disease is typically transmitted to humans through scratches or bites from infected cats and can cause fever, lymphadenopathy, and a characteristic skin lesion at the site of infection. Francisella tularensis is a small gram-negative bacterium that is the causative agent of tularemia, a disease that can be transmitted to humans through various routes including tick bites, contact with infected animals, or inhalation of contaminated aerosols. Tularemia can cause a wide range of clinical manifestations including ulceroglandular, glandular or pharyngeal, typhoidal, and pneumonic forms.
Ho, ho, ho!
Legionella pneumophila is a gram-negative bacterium that is commonly found in water systems, such as air conditioners and hot water tanks. Legionella can be transmitted to humans through inhalation of contaminated water droplets, causing a type of pneumonia known as Legionnaire's disease. This disease can be severe and has a high mortality rate if left untreated. Borrelia burgdorferi is a spiral-shaped gram-negative bacterium that is transmitted to humans through the bite of infected ticks.
Ho, ho, ho!
This bacterium is the causative agent of Lyme disease, a disease that can cause a characteristic erythema migrans rash, arthritis, and neurological or cardiac complications if left untreated. Now it's time for a quick quiz. Please leave your answer in the comment section below. What type of bacteria is Pseudomonas? Now don't forget to hit the like button and subscribe if you're not already. Thanks for watching and hope you check out future videos.
Ho, ho, ho!